In this presentation that is entitled Thermographic Analysis of the Evaporation Process in Surfactant Solutions. There were new non-invasive techniques for the characterization of biological materials. My name is Pedro Garcia, and I'll be with you for the following slides. Well, to start, it must be said that the infrared thermography, or ERT, is a non-destructive technique. This means that the technique doesn't have to destroy a sample to extract its characterization of it. Another examples are ultrasonic or electroresistance tomography, between many other techniques. Well, the RT works thanks to the Stefan Boltzmann law. This law states that the power radiance of a body is related to the temperature of this dimensional body. For those phenomena that occur close to the ambient temperature, it's like 300 Kelvin, the max length of a wavelength emission is around the 2 to 17 micrometers. This is the range of the electromagnetic spectrum. That is the infrared part of the electromagnetic spectrum. Now, due to this, the ERT is a very attractive technique to measure the biological phenomena. For example, to measure the changes on the ocular film. Well, our work proposed that the study of the thin films of aqueous solutions to mimic in some way the behavior of the ocular film. Now, to do this, we choose aqueous solutions of a surfactant. In particular, we choose the surfactant because its nature. A surfactant is an amphiphilic molecule. This means that it has two parts. The first, or also called the head, part of the molecule is hydrophilic. That means it tends to create a bond with water molecules. The second part of the molecule is hydrophobic. On the other side, the behavior of this part of the molecule is to stay away from the molecule of water. Now, this nature establish a very particular behavior of the surfactant molecules on aqueous solutions. When we put a certain amount of the molecules on the water, these molecules prefer to stay on the surface of the bulk, with the heads bound to the molecular waters and the tails up to the air or the interface. Well, also, these molecules tend to form onto ensembles. For example, a micelle. A micelle is a form to package the molecules of the surfactants, with the tails, quote unquote, hide inside the foam and the head rounded the tails in interaction with the water environment. Well, To study these interactions on the surfactants aqueous solutions, we propose the next arrangement. And this diagram, there is uh, this diagram explains our arrangements in four zones. In the zone A, we have our IR camera that measure the sample and a reference body in the zone B. The sample or reference body stays at a constant temperature thanks to our electronic control temperature device located in zone C. And uh, for the last, we have in our zone D a thermal bed where we put our, our source of surfactant solution at the same temperature or very close temperature to the temperature analysis of the sample. Well, in particular, we use a Xenix Gavi Plus 40 camera, ERT camera. This camera has the ability to have a good measure between 8 and 14 micrometers of the wavelength. Now, for the temperature control, this is a diagram of how it works. Our core in this control is an Arduino Mega board. This Arduino Mega board gives a PNW signal to a control driver that is fit for a 12-volt source and controlled by a PID control the response on a faulty result that stays the temperature of a sample constant. To measure the temperature of the sample and the reference body, we have two analogical digital converters. And for the last, to assure that the poultier have good behavior, we have a thermal interchange block with a work liquid flow and a thermal sink. This temperature control throws a pretty constant temperature with variation between 0.01 Celsius degree plus minus. Well, our work is divided in two stages. In the first stage, we try to determine the TMC of a well-studied sodium diacetyl sulfate aqueous solution. To accomplish this, we design the next sample container that we are seeing now in our slide. This sample container could retain a 1 milliliter volume aqueous solution sample. In comparison of the wavelength of the emission and the evaporation, this sample could be taken as a semi-infinite. Now, for the second stage of our work, we measure thin films on these aqueous solutions. We can now not use the same arrangement or the same sample container that we used in the previous stage. For this stage, we use another special container. In the diagram, the special container is exemplified by the yellow rectangle with the blue circles. This stands for a borosilicate slates with a layer of PFEP recoverment. Now, these PFE have a thickness around 200 micrometers. This is how we can create a very thin pools of aqueous solutions. Now, in the left part of the image, we have a diagram of the a scene that our camera sees, but in visible light. Our camera doesn't retain the information of the visible light. This camera translated this information into the information of the infrared light to a 16 bits resolution image in a grayscale, like we put on the part in the right part of the slide, mark it like with a letter B. Now, the temperature of our sample and the temperature of our surroundings are pretty similar. Our camera uh, throws a very low contrast image. Due to that, we need to determine to apply pre-process uh, pre-process image. Uh, sorry, we need to pre-process the image for the posterior study. Now, this is an example of our raw image thrown by the camera. As I mentioned before, have a pretty low contrast between our, our sample and our surroundings. To determine the sites where we are measuring our sample, we apply a subtle filter, like it could be appreciated in this image. Now, with contours well established, we indicate our size of measurement to follow obtain the coordinates of the region of interest or ROIs. Now, with these ROIs, we can now extract the intensity of the energy emitted by the sample and try to understand the behavior of this through the complete experiment. Now, our results. For the first stage, we generate this graph, the temperature versus relative emissivity, where the blue scattered dots are experimental measures, and the red solid line is a curve fit that fit on experimental measures. This fit follows a sigmoidal behavior with an inflection point around 25.4 degrees Celsius that correspond to the TMC reported by literature. Now, for the second part of our work, we measure the fluctuation of the emitted energy by our sample. This is a pretty distinctive behavior. In particular, we can appreciate two kinds of behavior of the sample. Two times close to zero